Am I audible?
Yes.
Okay. Good morning, students. So today we are going to continue with the Science 2 lecture, right? So before we start, I would like to tell you, please take your notebooks and pen with you, and everyone has to write down. And today what we are going to take, that is an important part of the chapter, right? And it is always most of the times asked in the exam, and you have to, and unless and until you understand, you will not be able to write. If you learn, if you read from the textbook, it will be difficult to understand. So please try to understand, and then you'll be able to solve, right? So we'll start with the chapter 16, heredity and variation, right? So in the previous lecture we have studied what is heredity, what is inheritance. We have also studied the chromosomes, types of chromosomes, DNA, the detailed structure of DNA. We have studied about RNA and the types of RNAs, right? So today we are going to continue with the chapter, and today we are going to learn the principles of heredity, right? Mendelian principles of heredity. So let us start. So the principles of heredity was first given by the scientist, that is Gregor Johann Mendel. So before we start with his work, what he has done, let us know about him. So he is the name of the scientist is Gregor Johann Mendel. He was an Austrian scientist, and he has done his studies on the inheritance of traits in the pea plant. Right? He was the first person to say that the characters are inherited from parents to offspring, right? And also he has given some laws, he has given some ways or the rules by which the characters are inherited from parents to offsprings, right? So and that is now forwarded in the study of genetics, right? So his work was not realized and recognized until the 20th century, but later when his work was discovered, then he is called as the father of genetics now. So I'll show you, this is the slide of a pea plant, right? Which you can see here. This is a pea plant, and most of the Mendel's work was done on this plant, right? What he say, what Mendel says is genetic material is transferred in equal quantity from parents to progeny, right? So the genetic material is transferred from one generation to another, and also it is transferred in equal quantity from both the parents. So what are the characters which are going to be expressed in the next generation, right? So for that Mendel has given the rules. Mendel carried out research in this direction and put forth the principles of heredity responsible for such inheritance. All of the Mendel's experiments were based upon the visible characteristics of the pea plant, right? So he has done his experiments on the pea plant. This is the pea plant. All of you know what is pea plant. Pea plant is the plant of vatana, what we say in Hindi, right? Yes, now what you can see in the slide is the dominant and the recessive characteristics in pea plants, right? So he has studied that some of the characters are expressed in the first generation, right? And the other character, though the character is present, it is not getting expressed. So the characters which are expressed frequently, that characters he said as the dominant characters, right? So in the pea plant, he has observed that he has studied the seed shape, he studied seed color. previous slide. Yes. So he has studied shape of the seed, seed color, flower color, shape of the pod, and color of the pod. Also, he has studied flower position and the height of the stem, right? So he said that the round shape of the seed, yellow color of the seed, purple color of the flower, inflated pod, yellow pod, and the axial position of the flower and tallness of the plant are the dominant characters, right? While wrinkled seed, green seed, white flower, constricted pod, green pod, and the terminal flowers and the small size of the plant are the recessive characters, right? So as we have dominant and recessive characteristics in plants, we also have dominant and recessive characteristics in human beings. Let us study a few of them. Yes, you can see here. So brown and black color of the eye, right? Brown and black color of the eye are the dominant characters, right? That means we will observe this commonly, while blue color of the eye is a recessive character. Then large eye, curly hair, dimple on the cheeks, right-handed person, then rolling of the tongue. You must have seen some of the people, they can roll their tongue. So that is a dominant character. Then being RH positive, that means we have the blood group. They are A positive, B positive. So being positive is a dominant character, while being negative is a recessive character. Then taste and normal pigmentation of the skin. So normal pigmentation is a dominant character, while albinism is a recessive character. Now this is important. All of you should understand this in order to write in the exams. So we are going to study the monohybrid cross. The cross with only one pair of contrasting characters is called as the monohybrid cross, right? So now in this experiment, Mendel has considered plant height as a pair of contrasting characters, that is tall plant and dwarf plant. Right. So what he has done is he has taken one tall plant. So he has taken tall plants and dwarf plants, and he has self-pollinated it. He has allowed to pollinate. Then the cross between tall and dwarf plant. What he has seen is in the next generation, all the plants which has grown, those were tall plants. Right now you can see the parent is tall as well as dwarf, right? Both the parents are there, but in the next generation he has seen that all the plants were, all the offsprings were tall, right? Then what he has done is he allowed the doing selfing, that is cross fertilization or selfing between these plants, the tall plants of the F1 generation, and he has found that in the next generation he was getting a dwarf plant, right? So he was very excited and he was very astonished with this, and he used to carry, he has continued with his work, right? So let us try to understand how this has happened. Yes, here you can see what he has done is in the parental generation, he has taken a tall plant and a short plant, right? This is called as phenotype. Phenotype means the character which is visible to you, right? So the tallness and the darkness of the plant is visible to us. So that is called as phenotype. Then the genotype is the factors which are responsible for the external characters but which we cannot see. That means he has said at that time, he has said, he has used the word the factors responsible for the external characteristic. But now we know those factors are called as genes, right? So the genotype, for dominant character we are going to use capital letters, and for recessive characters we use small letters. Always you have to remember whenever you are writing a dominant characteristic, you have to use capital letters and that too in pairs. And when you are using writing about recessive characters, you have to use small letters, right? So we are going to use, for tallness we are going to use capital T, and for darkness, we recessive character, we are going to use small t. Then the gametes of this. Now, if the genes are TT, obviously when the division is there, meiosis division takes place and the gametes become haploid, right? So the gametes will be, for tall plant, the gamete will be capital T, and for dwarf plant, the gamete will be small t. Now, in the first filial generation, there is capital T and small t. They combine to form the character, the genotype capital T, small t. Now, here you can observe the phenotype is tall, right? Even if there is one dominant character, the phenotype will be dominant, right? So, here you can see the phenotype is tall, right? Now you can see here. Now in the next generation, what he has done is he has taken the plants which he has grown in the first generation. So both the plants were tall phenotypically, but genotypically they were different. They were hybrid, right? They were not pure dominant plants. They were hybrid with the genotype capital T and small t, right? So now he has taken those two plants and he has done a cross between them. So the gametes of capital T and small t will be TT and capital T small t and capital T small t. Now, as there are four types of gametes, so the possibilities of the crossing are 16, right? So that we are going to see with the help of a square. This is called as Punnett square, right? This is the method how we write in order to find the characters. So here on the top of the table, we write the gametes for male gametes, capital T, small t. While here on the left column, we are writing capital T, small t of the female gamete. Now you have to do the combination. This T will take one T and one T of this. So you'll get TT. Then the next will be, so here you can see the next will be this capital T and this small t. Then you have to write this capital T of the male gamete and this small t of the female gamete. Then this small t of the male gamete and small t of the female gamete. Right? So you will get such four characteristics or four this. Now you can see here capital T, that means the plant will be tall, right? So, and that too, this is pure dominant. Both are capital T's, right? So, pure dominant, that is also called as homozygous. This plant will be homozygous tall plant. Now, here again, this is also a tall plant. This will also be a tall plant, but as it is having one dominant character, because the dominant character, it masks the character of the recessive, means it does not allow the recessive character to express. That is why it is called as dominant character. So as one T is there, the plant will be a tall plant, right? But here the genotype is different. It is capital T and small t, heterozygous. That means this plant is having the characters for dwarfness, but it is expressing the expression is a tall plant. This is also heterozygous, and this is homozygous, purely dwarf plant, right? Yes, so what he has observed is that this is important. Mendel crossed a tall plant with a dwarf plant and observed how the triads are transmitted the progeny and calculate the percentage of tallness and dwarfness in the subsequent generation. When a pure breeding tall plant was crossed with a pure breeding dwarf plant, that is small t, all the plants were tall in the first filial generation, right? Now this is important. Remember this, when the pure tall plant was crossed with a pure dwarf plant, all the plants in the first generation will be tall, right? Then what happened is when such an F1 tall plants was allowed to self-pollination, both the tall and the dwarf plants were appeared in the second generation, right? So in the second generation, the dwarf plants also appeared, right? You can see the results of the second serial generation. So according to the data collected by Mendel, out of he has planted 929 pea plants, 705 were tall and 224 were short. Thus, the phenotypic ratio of the plants is 3 is to 1, that is 3 tall plants and 1 dwarf plant. And the genotypic ratio is 1 is to 2 is to 1. That means, though there are 3 tall plants, but their genotypes are different. So, one is purely homozygous tall plant, but while the two are hybrid plants, that means they have the characters for recessive character, that is, they have the character of shortness, but still they are expressing a tall plant, and this is a pure homozygous recessive plant, right? Thus, it can be inferred that in F2 generation, phenotype are two types of plants, whereas genotypically there are three types. So I hope you have understood this much. I hope you have understood the Mendel's monohybrid cross. So if you understand the monohybrid cross easily, then you will understand the dihybrid cross as well, right? So now let us study without wasting any time. We will continue with the Mendel's dihybrid cross, right? Mendel brought about, now what is dihybrid? Now, in monohybrid, one contrasting character was studied. Now, here two contrasting characteristics Mendel has studied simultaneously, right? So, Mendel brought about a cross between a pea plant producing rounded and yellow seeds. You can see here, and a pea plant with wrinkled and green seeds, right? So he has taken a plant in such a way that the seeds produce, one plant used to produce rounded and yellow colored seeds, while the other plant used to produce wrinkled and green color seeds. So in this cross, two pairs of contrasting characteristics were considered: color of the seed and shape of the seed. That is why it is called as the dihybrid cross. Okay, now you can see the parental generation. The phenotype is rounded yellow seeds. Here you can see rounded yellow seeds, and the phenotype of the other plant is wrinkled green seeds. Right? We have seen what is phenotype, the external visible characteristic, right? So you can see this. So he has taken two rounded yellow seeds and wrinkled green seeds. Now, as we are having two characters, so we will have the genotype in this manner. You can see rounded. So for rounded, we are going to take capital R. So it will be capital R, and for yellow, it will be capital Y, capital Y. And now the recessive character. So we are going to take small r, and for green, it will be small y, right? So the gametes here are, now we know the gametes are haploid. So the gametes will have capital R, capital Y, and small r, small y, right? So now what will happen in the first serial generation? You can see capital R, small r, and capital Y, small y. In such a way, the combination will take place. So what will be the phenotype of this? Now, as the R is capital, the seed will be rounded, right? And as the Y is also capital, so it will be yellow. So, the next generation of the pea plant will be round yellow seeds, right? It will have round yellow seeds. That means dominant characteristics will be appeared, but the genotypically it is different, right? So you can see here in the first serial generation, the plants which are rounded yellow seed, they are having a hybrid genotype. See, can you imagine in that century, in 18th century, Mendel has found out that some characteristic is there, which is some is there that is a hybrid thing. He has found for the first time. He said for the first time, right? So this is a hybrid genotype. Then what happened is, now in the P2 generation, again the same way he has allowed the plants in the F1 generation to self-pollinate. That means the crossing takes place between those plants with round yellow seeds and round yellow seeds. Both are dominant characters. But now here the genotype was capital R, small r, capital Y, small y. Again here also that it was capital R, small r, capital Y, small y. And the gametes. Now you can see here the gametes. There are the possibilities of 4 types of gametes here. So you can see here, now you have to take one R and one Y. Okay, so what are the possibilities? Capital R, capital Y, capital R, small y, then small r, capital Y, and small r, small y, right? Once you write it down, it will be easy to understand, right? So then again here also the gametes will be capital R, capital Y, capital R, small r, capital Y, and small r, small y, right? So now we are having 4 male gametes and 4 female gametes. So the combination will give you 16 different possibilities. So let's see now. Yes, now you can see here. Now in your notebooks you have to make such kind of table. Draw the table like this, right here all the male gametes, and in the left column write down the female gametes. So the gametes will be capital R, capital Y, capital R, small r, capital Y, and small r, small y. In the same way for the female gametes also. Now let us try to write down the possible combinations. So here you can see, take this capital R, capital Y, and capital R, capital Y. So it will be capital R, capital Y, right? Then the next will be, you have to take this column capital R and small y, and here capital R and capital Y. So how will write capital R, two capital R's are there, and capital Y and small y. Then the next is capital R, small r. Here you have to take this column, this small r, capital Y, and capital R and capital Y. So are you following how to take? You have to take this column and this column. So it will be capital R, small r, capital Y, capital Y. Now here this column small r, small y, and capital R, capital Y, but you have to write in this manner: capital R, small r, capital Y, small y. Now here what it will be? Now you only have to say, make on your own, try to do on your own. What it will be? Capital R, capital Y, small y. First you have to write the R's and then the Y's, and first you have to write capital and then small. And then here what are the possibilities? So it will be capital R, small y, small y. Capital R, capital Y, small y. Here it will be capital R, small r, and small y, small y. Here it will be capital R, small r, capital Y, capital Y. In this manner you have to write down. Are you writing? All of you try on your own. You can see here capital R, capital Y, small y. Small r, capital Y, capital Y. In this manner you have to complete the table. Right? So there are 16 different combinations. Right? Now, before we proceed, now you only have to tell me which plants will have round yellow seeds, which plants will have wrinkled green seeds, right? So you can see here now, capital R, capital Y means this will be a round yellow seed. This will also be a round yellow seed. Round yellow. Here you can see it is a round green seed. Right, it is round, but it is a green seed because the recessive character has come. green color has come, right? Then this again will be a green seed because recessive Y is there. Now here you can see this is a wrinkled yellow seed. See it is a variation, right? So see how Mendel has told us about the inheritance that is inherited heredity and the variation. So here you can see the variation in the next generation. We were not having any uh, wrinkled yellow seed. We were not having the plant which is having a wrinkled yellow seed, but here this is a new characteristic. So this is a variation which has come in the next generation, right? Then again this is a wrinkled yellow seed. This is round green, and the last one is the pure recessive, that is wrinkled green seed. Now, before I tell you, so let me see if you can answer. You have to tell me out of this. You have to tell me the ratio of round yellow seeds and the wrinkled green seeds, as well as the what we can see. This is wrinkled yellow seeds. Sorry. Okay, so what will be the ratio? As we have seen in the Mendel's monohybrid cross, the ratio was 3:1 for phenotypic ratio, and the genotypic ratio was 1:2:1. Now, what will be the phenotypic and the genotypic ratio in the dihybrid cross? Can you tell me? Can you do on your own? Try in your notebooks. So you have to write the phenotypic ratio and the genotypic ratio. So let me help you. The phenotypic ratio will be like this. There will be round yellow seeds. There will be 9 round yellow seeds. Wrinkled yellow will be 3. Round green will be 3. Wrinkled green will be 1. Right? So there are 4 types of phenotypic characters, and the ratio is 9:3:3:1. This you have to remember. Always you have to remember, and this is same for all the dihybrid crosses, right? The ratio of the Wendel's dihybrid cross is 9:3:3:1, right? Can you see 9 plus 3 is 12, 12 plus 3 is 15, 15 plus 1 is 16. So 16 possible combinations. Now the next is the genotypic ratio. Now can you tell me the genotypic ratios? So the first genotype what we can see in the table is capital R, capital R, capital Y, capital Y. This is the pure dominant characteristic. You can see the genotype, it is pure dominant. This is hybrid. So there are 1 pure dominant, 2 hybrid, that is round yellow. It is round yellow, but the genotype is different. Then this is round green, right? Right, so there are 9. So we have seen 4 phenotypic characters, but the genotypic ratios, there are 9 possible combinations of the genotypes. So this is how the variation comes in the next generation. This is what is called as the variation, right? This was the work of the Mendel, which he has done almost 2 centuries ago, right? So he has found out that the variations are there, and the variations are there because of the factors which are present in our body or in our cells itself, right? So this was the person to say that some factors are there in the cell which are responsible for giving the heredity as well as the variation, right? So small r, small y is a pure recessive characteristic. So the first and the last are called as the homozygous characters, whereas the remaining 2 to 8 are called as the heterozygous characters, or they are also called as the hybrid characters. Yes, now this is also important. Most of the time it is asked in the exam. distinguish between Mendel's monohybrid cross and dihybrid cross, right? So these are the points. In monohybrid cross, inheritance of single allele pair, right? That means inheritance of a single uh, factor, that is, we call it as gene. Allele is nothing but a gene. Inheritance of a single gene pair. Here, inheritance pattern of two pairs of characters during the hybrid cross. Or in simple language, you can write here the inheritance of a single character, here inheritance of a pair of characters. Now the next is defined in Mendel's first law. This was the monohybrid cross tells you the Mendel's first law, while this is the Mendel's second law. Now this you have to remember, phenotypic ratio of the F2 generation in monohybrid crosses 3 is to 1, while the phenotypic ratio of the F2 generation in the dihybrid crosses 9 is to 3 is to 3 is to 1. Here, the genotypic ratio of the F2 generation was 1 is to 2 is to 1, while here in the dihybrid cross, the genotypic ratio was 1 is to 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 2 is to 1. So, this you have to learn. There is no other option. You have to learn the answer. Used to study the pattern of the inheritance during the first and the second generation offspring. So here this was done to study the pattern of the inheritance, and here also used to study the inheritance pattern of dominant and recessive characters. Now here basically we study the influence of the dominant characters on the recessive characters in dihybrid cross for the two different triads, right? So this was it. This is the differentiation between monohybrid and dihybrid cross. So before before I give you some assignment work, I would like to ask you if all the students have followed whatever is taught. I hope whatever I have taught is clear to you. But if anyone is having any doubt, you can ask me. Almas Miss.
First, give the assignment. Stop the
Okay.
recording.
Okay, this is an assignment to you. All of you write this down and try to do on your own. First, what you will do is at home, first you will read the Mendel's monohybrid cross, Mendel's dihybrid cross. You will draw the table which is given in the textbook, and then you have to do the assignment on your own. So the assignment is, the question is, explain Mendel's monohybrid progeny with the help of contrasting characters, round and wrinkled seed. So you have to take only this one characteristic. You have to do the cross between round and wrinkled seed by the Punnett square method. Okay. And the next is explain Mendel's dihybrid ratio with the help of any one cross. Right. So you can take any two characteristics out of the 7 mutual characteristics, and then you can do the cross and see whether you can do it or not. Right.